This is meant to be,
This meeting is being recorded.
this can be interactive, so I can't see you, but if you have any questions, feel free to jump in. As well, there will be a question or comment period at the end where we can have some discussion. And so in terms of the objectives for today, one, we're going to outline current challenges in public health bioinformatics. I think you know this better than anybody else, the audience on this call, so it's going to be brief. We're going to go through the BugSeq workflow, so I'll actually show you how users submit data, how those results look, and some brief interpretation. Then I'll overview some of our methods, specifically approach to pathogen agnostic and unified pipelines. Some open source stuff that we're working on because we really value the open source community and all of the work that you on this call are doing. And then finally discuss the future of bioinformatics in public health. So a bit about myself. Thanks, Logan, for the intro. My background is actually I'm a medical doctor specialized in medical microbiology, but I really love genomics. I've worked at genomics programs from the local hospital level all the way up to national levels in US and Canada. And so that's kind of how I got into BugSeq. And my background was actually in bioinformatics. Back in the day of microarrays, we were looking at gene expression in cancer. And then once I started specializing in medical microbiology, it was an easy transition. And so in this role, I've overseen thousands of analyses across all popular technologies, all labs from the local, again, to the national level, and a diverse array of pathogens from bacteria, viruses, fungi, and parasites. And we'll talk a bit about that today. So again, you know this, analysis costs time, accuracy, and health. So I think the golden, like what we would all dream of is we have instant results. So a user, which is your public health colleagues on the wet lab side, take a sample, sequence it, go after the sequencer goes into this magic box, and out the other end comes this analysis, which is easily interpretable by the epidemiologist, the microbiologists, as well as the other stakeholders that the public health lab serves. But this is hard. And it's hard because there's lots of open source tools, hard to get them working. There's also lots of proprietary tools, but you don't know what's going on under the hood and it can be hard to customize. If you're going to build an analysis for a pathogen, then you basically need to reinvent the wheel. So lots of public health labs right now are working on the same things, but not necessarily all coordinated on that effort. For some labs, compute infrastructure can be challenging. So for example, for our metagenomic analysis at BugSeq, we require servers with over 1 terabyte of RAM, and so it can be hard to achieve that on site. In terms of timing, some of the solutions can take days to weeks, whereas we know for PulseNet, for example, the requirement to turn around data is much shorter than that. And then especially once we start going into clinically useful results or infection control results, we really need to cut the time down to hours. And this is, again, serving stakeholders. And finally, when things don't go right, debugging can be painful and not fun. So this is just a general. The title is fairly ambitious, but I'll talk about really what we're doing today. And so if a lab sequences a sample, it can be a bacterial isolate, it could be direct from specimens. So for example, whether that's 16S amplicon sequencing or metagenomic sequencing, it could also be viral amplicons. They upload that data to the cloud. And so that data can be sequenced on any sequencing platform right now. We support all the 3 major platforms, Illumina, PacBio, Nanopore. And then the data gets analyzed in the cloud. I'll talk about some of the security aspects later in this presentation. But the first thing that we do is we detect, after quality control, we detect all pathogens in the sample. And so instead of having to do your traditional culture to figure out what a bacterial organism is or what a virus is, I mean, we do that online. And I know that most labs are still doing that today, but the future is we won't even need to identify things before sequencing them. The next thing is for bacterial or viral or parasitic or fungal, we identify antimicrobial resistance. Even if there's thousands of background microbes, obviously if it's an isolate, there's only one. That makes the analysis really easy. And finally, we characterize the organisms to track their spread in communities and hospitals. And so we look at, are these organisms related? Is this a hospital-acquired infection? How is this organism spreading in our community? And we really want to enable the public health labs to meet their mandate, and that's serving the EPIs, the hospitals, clinicians, and really with relatively fixed taxpayer dollars. Okay, so here's a quick demo of how it works for our users. So maybe I'll make that a bit bigger for you. Users will log on to BugSeq, and everybody has their own password protected login, but there's also the ability to share data across lab members. So your lab is a unit, and within that, there's different users. And then all you're going to do is you can upload a file. So I'm just going to upload test FastQ. You can upload, though, as many samples as you want in one analysis. You can upload paired end FastQ for Illumina. You can name your analysis if you want. Select the platform. So let's say we did some Illumina sequencing. Select the database. So for all of our public health labs, they're using our default database, which is based on high quality reference genomes online. As many of you are probably aware, there's also the NCBI NT database, which is the non-redundant set of GenBank. And the problem with this data set is a lot of it's open to user submission. And so there can be sequences in there that aren't properly annotated. However, it does encompass more organisms and has higher representation. So lots of BugSeqs, for example, environmental sequencing users use that NCBI NT. Then we'll just select a bacterial isolate, but if you're doing direct from sample or food, we also have those options. As well as for clinical samples, we specify the different types of clinical samples. For example, we know that an organism that's found in, for example, the CSF may be significant while it's not found. Well, all organisms, let's say, are significant in CSF, but an organism that's found in lower may be significant, but not significant in upper respiratory tract specimens. Finally, we support both DNA and RNA, and that comes into how we actually process the data later on. So looking at, for example, quality control, as well as assembly of the data, we use different algorithms for assembling DNA versus RNA. And then user would just go ahead and click submit. It's not done because I didn't upload all the files and fill in all the details. So then it runs, and our typical turnaround time is less than 4 hours right now. We're trying to get it down to less than 2 hours ultimately. And that's even for hundreds of samples, as we can scale in the cloud seamlessly. And then this is what I'm going to skip here. This is what the analysis results look like. So it outputs a bunch of HTML and PDF files, which are cleanly organized in the UI. The HTML and PDF mirror each other. And it's basically reports, because we want to tailor this for public health microbiology labs who are used to just having reports, where it's not this like interactive, kind of hard to archive thing. And so here's an example where 3 isolates were submitted. So there's sample 1, 2, and 3. And immediately off the bat, we see that, okay, they're all Klebsiella pneumoniae, with over 80% of the reads being classified as Klebsiella pneumoniae. The other percentage of reads, if you're curious, were classified to plasmid sequences in these isolates. These are real isolates from public health lab. Scrolling to the right here, we can see assembly length. So one of the first things we do after quality control is assemble the isolates. And so we look at how well they assembled, what percent of reads passed the pre-processing filter. So, were there too short reads, low complexity reads, low quality reads, etc. And then I'm going to scroll down and start going into the aggregate results. So this is a summary across all 3 isolates. So what plasmids are detected in each sample? For this, we rely on Mob Suite, which is a really fantastic tool, because I'll show you some of the information it outputs in a second. But we can then compare across all 3 isolates what are there. And I mean, these 3 isolates all have the same plasmids in them. But these are basically like stable taxonomic identifiers for the plasmid. And so if a user were to submit either the same isolate again or a different isolate with the same plasmid, this identifier would remain stable across analyses. And that's important because we know a lot of hospital-acquired infections, the antimicrobial resistance genes, are transmitted on plasmids. And so you might even have different organisms, but they may share the same plasmid. And so we can track that with this plasmid analysis. Next, looking at genotypic markers as well as phenotypic markers of or genotypic predictors of resistance, I should say. So we list out all of the genotypic predictors, including both genes as well as single nucleotide variants. And we can do that because we have the identity of each of these organisms. And so then we have a database for each organism, and then all of the single nucleotide variants in that organism that confer or predict antimicrobial resistance. And so we translate that the genotype into a phenotypic prediction of resistance, looking at the different common classes of drugs, and specifically reporting out drugs which would follow CLSI guidance, or for our European colleagues, the European guidance. So I'm going to just scroll down. You can see that these 3 isolates have the same AMR prediction, which is that they're fairly resistant, including resistant to ertapenem, imipenem, meropenem. So they're likely, or they are carbapenem resistant, but they're likely carbapenem producing organisms. The metagenomic assembly, I kind of highlighted that above. So I'm not going to go into, or classification rather, so I'm not going to go into that any further, but we look at what's in the sample. And we have 2 peer-reviewed published algorithms for metagenomic classification. One is a read-based algorithm, specifically focusing on long reads. And then the other one is assembly-based algorithm looking at short and long reads. And in both of our publications, we showed them to perform better than much of what else is out there, and external benchmarks have supported that. And I'll get into that a bit later. And then we dig down into assembly statistics, so the number of contigs, the quality of the assembly, et cetera. I'm going to skip through this. Read quality control. So many of you have seen FastQC plots. This is the same thing, looking, and these are all tunable for our users. So for example, some users say, oh, we want this amount of coverage, or we want this Fred score, and we can set the warning flags for that. Looking at each individual sample, they also get a report. And it will have additional details on that sample that's not included in the summary report. So for example, for each plasmid, we can look at what's the length of the plasmid, was it circularized, what are the number of contigs, and what's its host range. And this is pretty cool because when you have a mixed sample, so for example, if you had 2 isolates that were contaminated and sequenced together, you can actually predict which plasmid may belong to which isolate that was sequenced based on its host range. Obviously, if the host range overlaps, that may not work. And then looking at the AMR table, you can dive in deeper, looking at which genotypic predictor of resistance causes the predicted phenotype. So looking at, for example, NDM predicts, okay, ceftazidime avibactam resistance, or all of the 3rd generation cephalosporins and carbapenem resistance. So this really links that genotype and phenotype. There's additionally multi-locus sequence typing, Klebsiella for this one specifically, but we have organism-specific analyses for really hundreds of organisms now, and so that includes Salmonella, E. coli, Klebsiella, et cetera, as well as viral. And then there's some more details on assembly completeness, for example, benchmarking using single copy orthologs, and then depth of sequencing, looking at across the chromosome. So this is high level, just what we're outputting right now. And then there's also some outbreak investigation analysis. So our users can submit unlimited number of samples per submission. And then we will also aggregate their data across submissions. And so, for example, if a user today submits 10 isolates of Neisseria meningitidis, and then tomorrow submits 10 isolates of Neisseria meningitidis, we'll perform a unified outbreak analysis across those 20 isolates. So here's just an example, again, from a real user of 2 outbreaks of Neisseria meningitidis, or 2 clusters. And so what we've done, and this is going to be open sourced and put up as a preprint shortly, hopefully within the next month or 2, is we've developed a method that's similar to core genome multi-locus sequence typing, but it has some advantages in that we can generate cluster addresses which are stable. And so these cluster addresses reflect the allele distance between isolates. And so if they share the same cluster address, they're less than 5 alleles different from each other. But if the last digit of the address is different, so for example, if this was a 2 here, then we know that it's more than 5, but less than 10 alleles different. And so you can gather the relatedness of outbreaks based on their cluster addresses. The NM here stands for Neisseria meningitidis. And so users can continue submitting data, and their isolates will continually be added to their outbreak investigation with cluster addresses remaining stable. And then we also output a similar form, same isolates with a distance matrix. And so you've probably seen these both with allele differences or SNP analysis, where you can tell, okay, these isolates probably aren't related because they're 1,500 approximately alleles different, versus within this outbreak cluster, it's only 1 to 2 alleles or even 0 alleles different. So this is our general output right now, and there's more outputs. I just wanted to highlight these ones for what we're going to talk about going forward. Okay, so looking at some common applications that public health labs across North America are using us right now. One of our top ones would be AMR prediction, both for carbapenem producing organisms as well as for Mycobacterium tuberculosis. And this also is related to hospital-acquired infections, but we also serve hospital-acquired infections which aren't CPOs or don't have significant AMR like that, for example, VRE, MRSA, etc. Organism identification and metagenomics. This is an increasingly hot topic. And so organism identification, some labs are using sequencing to identify difficult to identify isolates. So for example, their MALDI fails, their traditional biochemicals fail, they sequence it. And so we can compare that against our database. Or if it's a mixed sample, then identifying all of the organisms and the pathogens in that sample. And finally, amplicon sequencing. So whether that's 16S, ITS, or viral amplicons. Just a couple of the names we work with. So on the industry side, we work with CDC, we work with all of the sequencing companies. We have a project with BARDA right now to develop a respiratory metagenomic assay. And on the kind of public health clinical side, we work with public health, we work with networks which power labs for millions of people. And these are just a couple of our close collaborators listed. Okay, so I think this is the interesting part now for this group, which is how do we basically string together all of this analysis? And the short answer, it's been quite a journey to piece together all these tools, both open source, but also writing a lot of the tooling and reporting ourselves to meet the needs of our users. And so the first question that we got was, how do you curate databases? And so many people think of a database as just putting together like a list of organisms which are significant or a list of antimicrobial resistance genes. But actually, what we want to highlight is curating databases is more complex than often conveyed. And so at BugSeq, we actually have one database for each of these functions, whether that's identifying the organism. So for example, you need reference genomes and you need matching taxonomy for all of those reference genomes, or whether that's antimicrobial resistance genes or variants and their predicted phenotypes, so linking that together, plasmids, MLST, etc. And so you need to maintain each of these databases. And then the other thing is the public version of these databases may not always be good quality, and I'll get into that in a second. And then version control is paramount. So what we see with, we know with public health labs and clinical labs is that we need to have reproducible analysis. So if you submit a sample today and you submit a sample tomorrow or in a week or a year, we're going to get the same result every time. And the only way to do that is by version controlling the databases and version controlling the analysis and actually linking together that analysis and database together. Okay, so at BugSeq, we spend a lot of time and person hours curating databases. And so here's kind of an approach that we've taken, or what we suggest to public health labs that aren't looking for a commercial solution, looking to do it all themselves from the ground up, where it starts somewhere. And so all public health databases have limitations. And so I just put these databases as an example for AMR. So there's NCBI's AMR Finder Plus, CARD, ResFinder. They're all really, I have to say, they're all great in the sense that, like, they've put in a tremendous amount of effort to put these databases together. And it's really appreciated both, I think, across, industry, academia, public health, the efforts that these groups have put in, and really the quality that they've gotten to. And then I'll caveat that there are obviously some limitations, like with everything. And so even with these databases, ResFinder is the only one that will link specific drugs to each gene. NCBI and CARD will just give you classes of drugs, which is already a limitation because it's not really that helpful to say that beta-lactams are resistant when there's so many drugs within beta-lactams. And so even within ResFinder, though, they'll only link what's published in the literature. However, if a group tested an NDM only against one drug and then published that, the NDM will only be annotated against that one drug. So not really representing the depth of resistance conferred by each gene. The next is building a multidisciplinary team. So it really wouldn't be possible to improve on these databases or curate them further without having microbiologists who really know the nitty-gritty about each gene and what resistance they confer and the classes of resistance genes, as well as having that kind of bioinformatics side to actually implement that in the database, translate that into the analysis, validate that it actually works, maintain that version control. And so one approach is to take these existing databases, combine them, find errors, add missing data. And then once you apply that to thousands of samples, as we've done, then you can start incorporating all of this diversity that you see across your users. So for example, we'll see repeatedly errors in the database because a user will submit a carbapenemase, but then it's not annotated as resistant to all of the carbapenem drugs. So then you can go in and correct all of that. But then the next time, say you correct it for NDM, the next user will submit a KPC and then it may not be annotated across. And so by incorporating that feedback from our users, as well as some of their actual lab feedback, so they'll say, we did this test in the lab to prove it's carbapenemase, here's all of our evidence, you can refine the database such that it keeps performing better and better. And so a bit about our curation, we have over 300 users of BugSeq. Not all of those are public health. They span the spectrum of public health, clinical, and then there's an academic kind of translational group as well. We process over 3,000 samples, and so all of those samples have contributed to the quality of our database. And then we have a dedicated lead to review the literature, curate the database or databases, and thousands of hours have gone into this. So here's some examples. So I mentioned the classes of drugs. So if you look at AMR Finder Plus, it just reports NDM1, which is a common carbapenemase, as resistant to carbapenems, but that's all, it stops there. If you look at CARD, it reports it as resistant to penems, carbapenems, cephamycins, and cephalosporins. But then in our database, we've gone ahead and annotated that to each drug within each class and incorporating some unique for each enzyme. So for example, for NDM, we know that they're resistant to carbapenems, cephalosporins, et cetera, but they're not resistant to monobactams. And so you'll notice that aztreonam is missing from our result here. Whereas if we were to look at some of the ESBLs, for example, aztreonam would be included in that list. So like CTXM. Another example of our curation of taxonomy. So if you look at, say you're going to use NCBI as a base database for your taxonomy, there's actually 191,000 assemblies of E. coli on NCBI because everybody is sequencing E. coli for PulseNet, for outbreaks, et cetera. And so then the question is, are you going to include all of these assemblies in your database? There's obviously pros to including such a large number. For example, you're going to capture the entire pangenome of the species and all of those rare genes that aren't really, part of your core genome or common. However, there's a lot of redundancy in there where the core genome of 1,500, 2,000 genes, however many it is, will be shared across all of those E. coli. And so one approach that's been taken in the literature is you include a representative sequence. So you only take your reference genome of E. coli. Another approach that's been taken is you take only the unique sequences. So you'll line them up against each other, which is very computationally intensive for 191,000 genomes, and then only take the parts that are unique. And then finally, you can just take all of them. And then there's some algorithmic tools that you can use to speed this process up, whichever approach you take. Another example of taxonomy curation is metagenomic detection of SARS-CoV-2. Or this is just one specific example, where many of our users are now doing respiratory metagenomics. And so if SARS-CoV-2 as currently annotated in NCBI is annotated under the species severe acute respiratory syndrome related coronavirus. And so if you report out at the species level, then this is what you will report out. And users will get very alarmed that there is SARS in your sample, but not SARS-CoV-2. And so SARS-CoV-2 is actually annotated at the no rank taxonomy, which is below species, but is not the strain level. And so you have to know to specifically for SARS-CoV-2 to report out at the no rank below the species. That's just one example. There are many other examples. And here's an example for plasmids. So if you're doing nanopore sequencing, the Oxford Nanopore Rapid Sequencing Kit is contaminated with a plasmid used to produce the transposase. And so we actually find that in all of our samples from users using this kit. And so we know immediately when a user submits data and we see this plasmid, oh, they're using the Rapid Sequencing Kit. And so that can be confusing to users to see the same plasmid in all of their samples. However, if you annotate that properly and label that for your users, then it will help them understand the data and why they're seeing this plasmid, and that it's not necessarily unexpected. Okay, next looking at how to build pathogen agnostic analyses. So the traditional paradigm that we've seen across public health labs and bioinformatics is let's build an analysis for X pathogen, whether that's SARS-CoV-2. So you can think about like Arctic analysis, or let's build a different analysis for another pathogen like our enterics. But we need to zoom out and think about how do we build one analysis or a set of analyses that scale broadly across organisms within, let's say, the same kingdom. So how do we build an analysis that scales to all bacterial organisms? Because that's really what's sustainable going forward and will give you the bang for the buck. And so your lab, we know your lab is expanding sequencing, and I already mentioned the PulseNet COVID-19. But some of the limitations as well is if you implement multiple tools, for example, one for bacterial analysis, one for viral, or even within bacteria, one for TB, one for E. coli, one for Shigella, you get this fragmentation, complexity, and suboptimal solutions. So you may use even a different tool to pre-process your reads for one pipeline than another pipeline. And that may stem from multiple people working on different pipelines as well. And then if you were to use a pathogen agnostic approach, you could say, hey, we're going to use Trim-O-Matic or we're going to use FastP for all of our pre-processing. And so all analyses will share that same base of code, and we can continue updating that together. And so that's what we do at BugSeq, where we take this pathogen agnostic approach. And so here's one approach that we've taken to solving this. So there's a question of what's in my pot. And so these can either be reads or contigs. It doesn't really matter for this example. And in the literature, people have taken both approaches where either binning reads or contigs from assemblies. And so then you can bin them by species, you can bin them by strain, or you could do unsupervised binning, which bins them by patterns in the sequencing data. So for example, tetranucleotide repeats. At BugSeq, we particularly like binning by species because it's fairly high yield, and that's what our users generally care about. However, there are some exceptions to our binning process where we'll, for example, bin by serovar for Salmonella. But once you have your bins, you can then go ahead and operate on each bin. And so you can characterize the genome or the reads inside of that bin. So for example, for Salmonella, we can serotype and say, hey, in this bin, there's a Salmonella typhi. And that's from the original pot where it's all mixed together. Or for the E. coli, you can do AMR typing on or testing on or prediction. So you can say, hey, this looks carbapenem resistant. And for the Klebs pneuma, you can do sequence typing. These are just examples, but you can do whatever analysis you want on each bin. And so that's exactly what we did in this publication in Nature Communications Biology, where we built a metagenomic assembly binner to operate on each bin. And so the general concept is you bin the assembly to taxa via Minimap2 alignment to a database. So we have a database of all these high quality reference genomes. And then we use Minimap2, which is a really good aligner for long reads as well as assemblies to align to that database. Then based on the alignments, we bin by the taxa that they align to and then operate on those taxa. It's as simple as that. There are some added features. For example, how do you handle plasmids if you don't have them in their database, which we deal with as well. But the general concept is just assigning contigs to where they align in the database. And so here's some data from that paper where we first applied it to an isolate of Bacillus anthracis, the etiologic agent of anthrax. So this was sequenced at the CDC. Actually, they care a lot about anthrax, obviously, as well as across the LRN. And so we identified it as Bacillus anthracis in 1 hour and 20 minutes of analysis. And this is based off of nanopore sequencing, I should mention, and assembly. MMSeq2, which is a commonly used tool for taxonomic assignment of contigs and assemblies, could only assign it to, but I'm not even going to pretend to pronounce that. And then Diamond and Megan LR, another commonly used tool, could only assign it to the genus level at Bacillus. You can see as well that the runtimes were drastically different. We applied this to some benchmarks. So for example, we know that the CAMI benchmark is widely used to demonstrate the power of binning tools. And we showed that BugSplit could significantly improve on the existing taxonomic binners. We don't look at unsupervised binners because we consider that a different challenge. And so, for example, you can see that the number of bins that we had, which were complete or not contaminated and very complete, is much greater than MMSeq2 and Diamond. And as well, if we look at this plot, the completeness of our bins was significantly higher than Diamond and MMSeq2 while maintaining the same purity of the bin. So completeness is how much of the bacterial genome you actually recovered. So ideally, you recover 100% of the genome in a bin. And purity is how much of other strains or species did you put into that bin by accident. And so ideally, you want 0% contamination or 100% purity, so that you're not putting in mixing organisms together into the wrong bin, because then in your downstream analysis, you may end up saying this E. coli is resistant to X drug when it was really a different organism in the same sample. And so we then applied this to some clinical samples. So we applied it to blood cultures spiked with Klebsiella pneumoniae, and we showed that BugSplit was the only tool able to accurately recover the Klebsiella pneumoniae full genome directly from the blood sample sequencing. And then on that Klebsiella pneumoniae genome, we could then predict AMR and serotyping again, as compared to the completeness of MMSeq2 and Diamond, again, which were much lower and did not provide the necessary information to get accurate AMR serotyping. And we also applied this to other examples like urine for Neisseria gonorrhea. So we could predict AMR directly from the urine sample. And we showed that we can predict novel pathogens like novel viruses, for example, for the next pandemic directly from respiratory samples. So that was the concept of this paper. I'm going to speed along to enable our discussion at the end, but just looking at the outbreak analysis, which we're providing to users, I said that it was similar to core genome MLST. And so traditionally, I think you know this better than most, is that core genome MLST or whole genome MLST are the taken approaches for bacteria, or SNP analysis is also a commonly used approach. Some limitations of these approaches are that you need a scheme. And that scheme requires hundreds or thousands of genomes to build, especially to be stable. Requires curation. There are some open source tools as well as proprietary tools to curate these schemes. And then you generally need a centralized naming server to house that scheme, download it, keep it updated, etc. That's all pretty complicated. Some limitations of SNP analysis. So generally does not consider genetic recombination. You can, but then you can't compare analysis across samples is what we found. So in general, doesn't. And then SNP analysis, it's harder to share data, specifically around people get all up in arms about privacy laws. And so that can be a limitation there. So what we've done is we've implemented an approach which we call reference-based MLST or RefMLST. And this is combining the advantages of core genome and SNP analysis. So we don't require a scheme. Basically, the genomes are compared based on the genes found in the reference genome, and we exclude mobile elements and plasmids, so we don't have to worry about those. You don't need a centralized server, so we actually do this distributed, where alleles of genes are compared based on the hash instead of a naming for that. So you don't need the server to store, say, AAA equals allele 1 versus ATA equals allele 2. You just hash that sequence and compare the hash. And this enables reproducible allele distances between isolates and that cluster addresses that we showed earlier. I'm not going to dig into this too much further. I already showed you that because a preprint will be coming up shortly on that method. And then finally, I want to say that we really appreciate all of the work of all of the people on this call, as well as the open source community. And we stand on the shoulders of giants. We wouldn't be here without some of these contributions. And so we want to give back to the community. And so here are many of the tools, or here are a list of tools where we've contributed pull requests on GitHub. And our list is actually bigger because obviously not all code is on GitHub, but some pretty common tools that we use and that we think are valuable to the community, like MultiQC, Nextflow, ResFinder, etc. Okay, looking at implementation of BugSeq, a lot of people ask about confidential information. And so one of the things that we say is, and this applies really to any lab, where sample names can be any unique string. So don't submit personally identifying information. That's on the submitting labs. Metagenomic data can contain human sequencing data. So for example, even if you use the best host depletion methods in the wet lab, you're still going to get some human sequencing data in your sequencer output. And so we've actually open sourced a tool to scrub human sequences from sequencing data. I've linked it here before upload to our cloud, but this can really be used with any service. All it does is it maps the reads against a reference genome using Minimap2 and takes the reads that don't align. And then we encrypt all of our data in transit, REST, firewalled servers, etc. In terms of data retention, so we ask our users what they need. Many of our users say 7 years. Some of our users have said 20 years or more. We can do that. I mean, we store our data on Amazon, so Amazon is going to be around in 20 years, and it's really to meet the needs of our users. We process data in users' jurisdiction. And so many users will say we can only process data either in the States or even our state or region. And so we can process data where Amazon has servers. And then we have robust access controls to follow regulation. In terms of getting labs onboarded, there's sole sourcing. So that's a whole process, government contracting, etc. We've been through this all before. So we know the hurdles about it, and we can help labs get onboarded quickly and get funding to support this work. And so just looking towards the future, we know that public health labs are moving towards a future where every sample that comes in the door is going to be sequenced, whether that's your run-of-the-mill TB, whether that's a virus, or whether that's something completely new and novel. And so this is really going to enable improving the health of patients and populations in our communities. And so we need to do that with rapid turnaround time and with precision. And that's what we're going for at BugSeq. So enabling rapid pathogen detection, AMR, and outbreak typing. And finally, taking back to the bioinformaticians. So we know that you will be the data champion. So there's probably not going to be any person more important in the lab in the future of sequencing than the bioinformatician who holds all that data and knows how to process it. And so we want to help you start building your toolbox now with some of our things that we've built. So if you're interested, you can drop me a line. I'm happy to chat offline. And I'm also happy to answer any questions now. And so I'll thank you for your attention and open it up to you.
All right. Well, thank you so much, Sam. That was a fantastic talk. I will go ahead and open it up for questions now. I guess I can throw it out to the audience for questions first. As always, I have a couple of questions prepared myself, but I figure I'll give everybody else a chance to ask their questions first, because it's a lot of information, and I think that there's probably going to be people who want to start some discussions. It looks like there's already some questions in the chat. Could you post the link to the host scrubber in the chat?
Definitely, yeah. You'll have to give me a second to pull it up. I can do that.
Yeah, I'm curious about that. Did you use something? I mean, I guess if you're just kind of like aligning to a host genome and then taking the reads that don't align, there's like precedent for that before. But I was wondering, did you get that from like NCBI, or have you checked it against any of the other human read scrubbers?
So I should say that currently it's tailored for long reads. However, it can be easy. Minimap2 can easily handle short reads as well, as I'm sure you're aware. We haven't compared it against any of the things because no tools that we saw out there did effectively both long reads and short reads with high sensitivity and specificity. Minimap2, like credit to Hang Li, the tool is fantastic. And so like sensitivity and specificity and speed. And so this is really just a tuned approach to leverage Minimap2 to filter reads.
All right. Well, while people are thinking of questions, I guess I will kind of go ahead and ask some of the other questions that I have. So with all of your databases, they're all, it looked like it was all in-house curation. So like you guys are curating these databases and keeping them up to date. Is that, I mean, is that's true right now for BugSeq?
Yeah.
Wow,
Yeah.
that's a lot of work.
Yeah, so we have a team who's really dedicated to updating databases because we see that as a critical need of the community. And so looking at taxonomic identifications, AMR, I would say, is the big weakness out there right now, where there's no strong AMR ones. I see somebody has commented, are these, so yes, if you actually look at ResFinder, GitLab, not GitLab, Bitbucket, they have been submitted upstream. However, the upstream maintainer has been slow to incorporate them, but we have submitted them upstream. And so we do want to contribute them back to the community. Not all of them will be contributed upstream because not all of the databases are open source, like ResFinders, for example.
Are your databases open source?
So they currently aren't, and that's because we are working to maintain the version control with our analysis tools. However, eventually we do plan on open sourcing a lot of our databases.
It looks like there's another question in the chat. So sequence data you used are mostly ONT platform. Would Illumina sequences result in similar analysis performance, especially when dealing with metagenomic samples?
Yes, is the short answer. So we, as part of our bug split paper and cited earlier, we applied it to the CAMI dataset, which is Illumina sequencing. It's the most commonly used benchmarking dataset for metagenomic classification and performed better than all other tools, I believe, in our benchmark on that dataset. And then we've seen across hundreds of users who have submitted Illumina data that it performs better than most other tools out there. And we think that's because you can get a lot more information via assembly than you can via K-MERS. And so when you have the really, hundreds of thousands of base pair contigs, it's much easier to classify them than 100 or 50 base pair read.
More question. Can you read the questions in the chat, or?
Sure, yeah.
Like
so the next question is jumping off Logan's question about database being open source eventually. What part of your code is open source and where can we find it? Will your code eventually be open source if it is not? And so yes, the goal is to open source large parts of our code. Some parts of our code are open source already. So that key bug split taxonomic binner is open sourced attached to that Nature Communications Biology publication. So if you go into the supplementary material of that, you'll be able to find the code. The host scrubber, I just saw linked in the chat. Some of our nanopore things are open source, like pore chop. So the ability to, this isn't us, but we built off of it or forked it. And so that is open source, the ability to trim nanopore adapters. And there are a couple other pieces that are open source. And our goal is to continue contributing back to the community because, again, we really see value in that.
Awesome. I love to hear that. One thing that I had questions on, because I saw it kind of come up in the sequence comparison, like between samples and when you're looking at like outbreaks and things like that. So I know that PulseNet has had like a lot of trouble with the kind of address, like the genomic address sort of thing, where you have, some sort of scheme in order to bind them together. But you said that yours was stable. So could you explain that a little bit more? Like how have you ensured that your scheme is going to stay stable?
Yeah. So there's a comparative approach which is called SnapperDB, which is based on a SNP analysis, and so they use a reference genome. And so if you continue mapping to that reference genome, then your reference is stable. So similar for us, we use a reference genome to define where the genes are located or what the gene sequences are, and then can pull all of those gene sequences from the assembly that we're examining. And so by using the same reference genome every time, we get the same gene sequences every time that we pull out, and then we can hash those. The hash is stable from that isolate, and then compare that across isolates, leads to the same differences every time. So that's briefly how we can compare stably.
Interesting.
I see another.
Yep, in the chat, you've got it. Okay.
Thank you. Can BugSeq access the data directly from Basebase or cloud sources? Do we need to upload sequence data manually? So currently, there are a couple ways to get data into BugSeq. One of them is our web UI. That's the easiest one for users who are just looking to get their feet wet. And I should mention as well that we're offering 3 free trials for users right now in Public Health Lab. So if you want to go onto our website, you can just register and try it out yourself. The other way is via API access. So we actually do have API, and some of our users like CDC is using our API. And the third one is custom integration. Yes, I can post the code afterwards. So we actually integrate with some EMRs right now, for example, LIMS Lite, but we do not currently directly interface with BaseSpace, but that is in the works.
All right. And so I had another question, but I think you already answered it. So the back end kind of cloud computing that you guys are using is through AWS. Is that correct?
Yeah.
Okay. If users had an AWS account already, would you be able to kind of like utilize your own, like hook it up to BugSeq somehow and utilize your own resources, or do you have pretty much your kind of like architecture and setup?
The latter.
Okay.
So getting like, your tooling working on AWS can be incredibly complex. And so unfortunately at present, we can't interface with Lab's existing AWS infrastructure.
That makes a lot of sense.
And I see another question. Our Office of Privacy and Security has pretty much said no to PAAS. Is there an enterprise local BugSeq option? So not currently, however, it is in the works as well. A lot of these things are in the works. So we know, especially in clinical labs, that sequencing will have to happen on site. And so we are working with some of the sequencing manufacturers to make that happen where it's on device. In terms of your Office of Privacy and Security, I can't comment too much about them, but I would challenge that notion where data can't be uploaded to the cloud, because how are you going to scale genomics in the future? I think it's going to be very, very challenging to maintain all of your compute on premise. And I think also there's people who can generally do it much more effectively than public health labs, both maintain privacy, security, and scaling with cost effectiveness. So for example, Amazon is much better at that than most public health labs. And so I would challenge that notion. One thing that we commonly ask our users is like, where is their credit card data being processed? And I know this isn't you saying no to it, but like all of your credit card data, all of your banking is in the cloud. And so it's surprising that like bacterial organisms with no human data, no PII involved, their sequences cannot be uploaded to the cloud. And I know there's lots of caveats around that, but it's just like, to get your thinking going. And then thanks for preaching to the choir. Cool. And then in terms of any plans to include plasmids, mobile genetic elements in the ref MLST method in the future, there are examples where whole core genome MLST doesn't work well due to clonality of certain organisms and serotypes. Sounds like a really cool approach. Thanks for sharing. That's a great idea. I don't think we had directly considered that yet. To be honest, the mob suite method I'd highly recommend if you're looking at plasmid typing is quite effective at having unique taxonomic identifiers for plasmids. However, if you do need kind of more nuanced or tailored approach for plasmids, yes, I suppose looking at their gene content and comparing that across plasmids would be an interesting way to do that.
So one other question that I had was, you said that you have kind of a wide basis of users. It spans from kind of like public health all the way to academia. And so is this being used in clinical labs by like physicians and hospitals and things like that? And how easily has it been to integrate this into their kind of like existing workflows and systems? I'm just kind of wondering about like, what is the barrier to like training and how well has it
it's a
been
large barrier. It's a large barrier, but we see it's coming regardless of what we like. And in public health, we need to support basically the users that we serve to also do that while still getting the data, because the clinical users, they don't necessarily care about all of the public health implications. They have different interests. And so it's how can we support them to also get that data back and make the data as the powerful tool. And so some of the barriers I can highlight for the clinical labs span everywhere from having the technical capacity to prepare samples, to sequence it. You know, sequences aren't very common in clinical microbiology labs, to the data analysis, which we're trying to solve for them. And then from a regulatory perspective, like doing this as an LDT can be complicated because there's a large barrier. You need to validate that it's working, meet all the CLIA regulation. And so that can be a challenge too. We do see this changing where, for example, we know that the FDA is going to be coming out with, likely coming out with NGS-based diagnostic guidance. There will likely be FDA-approved NGS diagnostics in the future. I can't say when. And so this will be getting easier and easier. And so it's important for clinical labs really to get started early so that they're ready for this technology to come to them. In terms of, can 3 free trial samples be from any source? Yes, so you can submit Illumina, Nanopore, PacBio, can be really metagenomic, bacterial isolate, viral, whatever you want, give it a go, and happy to see how your results turn out. I don't know if that's what you meant by any source. Yeah, so we don't operate on a cost per bacterial genome. The reason being is that doesn't really align with the value of our users. So BugSeq's on a subscription model, priced annually. And if you're curious for pricing, then get in touch with me afterwards.
What does the customizability of the reports like? Like, would users be able to kind of modify those themselves or pick and choose kind of like different things that they wanted to see? Or if they wanted to like add something, would they be able to kind of like, you know, put their own spin on things?
Yeah, so I mean, we don't let users go in and actually like change the code, but we're open to user suggestions to really change anything in their own personal results. So I can give you a couple examples. So BugSeq right now powers anthrax analysis as part of a pilot program at the CDC for bioterrorism. And they obviously are very concerned with detection of genetic engineering, as well as the reporting so that LRN members can interpret the results. So for example, they said, oh, we want an executive summary at the top saying whether you need to phone the CDC or not about the results. And so we could easily put that into the results. And that's based on, you know, whether there's AMR detected, whether there's plasmids detected, et cetera. And so we can take what our users want. Another one that we've heard from users is the CDC, for example, may require 40x coverage for reimbursement of the sequencing for hospital-acquired infections. So we can then put a flag into the result saying, is this, are all of these isolates 40x coverage or not, so that it flags you like, hey, we need to resequence these isolates and get more coverage. So there's lots of customization that can be done, and we love to work with labs to achieve their needs.
Awesome. I think that was most of the, oh, we got another question in the chat.
Yes, so the short answer is yes. Some metagenomic analyses, especially like if you're doing massive samples, really deep sequencing, assembly, metagenomic classification can require over 1 terabyte in RAM.
And so the organisms that are supported through this are basically, it sounded like it's mostly bacterial is what you've been focusing on, but there's also potentially viral and other maybe like eukaryotic organisms as well. Is that in the works or?
That's accurate, yeah. So we know that, you know, any kingdom of life causes public health problems. So I'm not talking about humans, but I'm talking about like Candida auris, for example, from eukaryotes, is a really hot topic in public health and also has its own unique challenges. And so we are building specific eukaryotic analyses in there. But the general kind of framework, you know, if you're going to do assembly and classification, that can work on whether it's fungal, bacterial, parasitic, which we have some users, and viral as well, which we have some big projects on.
Awesome, thank you. So I do want to be cognizant of people's time. It's about 2:59. So I am going to go ahead and stop the recording. If I can find